Thank you for giving me the opportunity to talk today. So I'm going to be talking about hybrid organic inorganic chromophores, and I represent one of the researchers part of AUCAOS from New Zealand. So if everyone's forgotten where that is, it's just off to the right in Australia. We make jokes that you're the West Island of New Zealand. We have the North, the South, and the West Island. Wellington in summer looks like this. At the moment, it's really sort of quite miserable, but I've been here for about 2 years and I'm starting my own lab effectively on luminescent materials for solar panels, LEDs, and solar concentrators. So this talk today is going to be about light harvesting systems. Just briefly, a bit of motivation. Why do we want to harvest light? So effectively, the sun puts out an enormous amount of energy, and nearly all life on Earth can be traced into absorbing sunlight in some way. So this is a picture of the Amazon rainforest, and just all that biodiversity and everything comes from absorbing sunlight. And so since life has thrived on absorbing sunlight, it's created very highly efficient, but also very complicated ways to channel this energy into different uses. So this is our photosystem 2, and that's what is used in photosynthesis. And this is a bacteria phycobilazome. And what they are is that it's large protein manifolds of basically chromophore proteins that absorb light and funnel it into a central unit or direct it away spatially to allow it to do work somewhere else. And so why would this be good? Effectively, if we can mimic nature, we can then either create things that harvest light better or emit light better. So our LEDs, our solar panels, lasers, solar concentrators, anything that needs spatial or energetic control of light, we can learn from these light harvesting complexes. And so I haven't given this talk before. I don't know if you saw at AUCAOS last year, I was sort of building up into attaching chromophores to nanocrystals. I was just looking at chromophores. So we're going to see how it goes. And so the first thing, if it lets me choose, what are our sort of building blocks? So we can either use organic chromophores to build these light harvesting systems, and they're really good because they have high absorption coefficients and they're quite bright. So you can easily get PLQEs sort of up to unity, 100% in some of the good dyes. The problem though is they're quite unstable under UV light because they are just conjugated bonds. You put a bit of UV light in, you can break a double bond and you can break your molecule. They do have small Stokes shifts, so that means they have high levels of reabsorption, and the tunability isn't the best in that every color effectively needs a single different molecule. You can extend your chromophore systems to change your color slightly, but all the best dyes are all completely different class of molecules. This is a perylene, which is red, this is fluorescein, which is green, and this is a coumarin, that's blue. So if you want all the different colors to start building these large manifolds, you need to synthesize completely different molecules, so it's quite hard to tune. The other option is inorganic chromophores, which again have high absorption coefficients. They're tunable now due to size and composition. So these are cesium-lead halide nanocrystals where the iodides are red, the bromines are green, and the chlorides are blue. So any color in between is just a ratio of those halides. And they're also very stable. And then other chromophores are quantum dots. You can change these colors by changing the size. So instead of synthesizing all new molecules, like here, you can synthesize just different sizes of quantum dots. The problem is the best ones are toxic, and the ones that aren't toxic require these complicated shells to have high efficiencies. So you take your core nanocrystal and you grow passivating layers on the outside, and that's going to increase the complexity. So when we look at how to make light harvesting systems out of these solely, you can see that making this molecule here, which looks like a link between a bunch of perylenes and some other chromophores, is a really complicated synthetic process. And then if you look again at the inorganic system, as you start layering more and more different energetic cores, although you can create these sort of energetic manifolds, the complexity again is a problem. So what I'm looking for is a simple solution where we combine both. And so I've termed these hybrid inorganic chromophores for light harvesting applications. And at the moment, I've got maybe 3 or 4 projects based on this idea. But essentially what you do is you take a nanocrystal and you replace the ligands around it that give it colloidal stability with something that has an energetic property. So you can either put energy from your outshell ligands into your nanocrystal or take energy from your nanocrystal into your ligand. And the idea being then you can build these quite simple antenna complexes by wrapping your nanocrystal in different ligands, or you can do things such as singlet fission or up conversion because the nanocrystals have high spin orbit coupling, so that it can emit or transfer triplet states quite easily. And you can also do things where you can wrap, we've recently wrapped nanocrystals around a bit of glass ball, and you can get a whispering gallium mode laser from these sort of systems as well. So there's a lot of potential there, but we're only just sort of scratching the surface and starting. So the potential here is effectively any nanocrystal you can find. So your lead chalcogenides, your mercury-based or your perovskite-based, cadmiums, indium phosphide are all the sort of crystals we make in my lab at the moment. But there's plenty more nanocrystals we can choose from. And then you just look at your linker on your ligand. You can change your binding group to have sulfurs, carboxylic acids, amines, these disulfur groups here, trisulfurs, like thiols. You can have a linker, which changes the distance between your chromophore and your nanocrystal, which would change the energetic interactions. So you can get more or less fret or energy transfer. And you can also change the energy of your chromophore. So looking at here, these are just our current ligands. I think we're up to about 23 now, but I didn't have time to draw them all. But we've got different chromophores here. We've got amphrosines, paralenes, tetrasines. We've got different linkins, thiols, carboxylic acids. And we've also got different chain lengths as well shown here. So we can do a lot of different things, interact them with any ligand or any nanocrystal. What I'm going to be talking about today is just two simple cases, which we've just submitted for publication using parylene diamides and an amphrosine and attaching it to cesium lead halide perovskites. And so again, the color of these nanocrystals is dictated by the halide composition. And these are really simple to make. You just take basically a cadmium olite and you inject that into a lead solution and you can make, or a lead halide solution, you can make these nanocrystals. So the first case we're going to be looking at is what happens if we start with energy transfer from the nanocrystal into the ligand. So for this, we're going to be using this parylene red, we'll call it. It's just a parylene diamide with base substituted phenyl groups, and then this parylene orange here, which is just the standard parylene diamide core. And they've got this sort of long linker with a carboxylic acid on the end, which will bind to our nanocrystals. And we just chose these molecules mainly because we had them around, and also because we can make a cesium lead bromide nanocrystal that's higher in energy. So see here, we've got higher in energy nanocrystal, it's sort of a green. We can then energy transfer into our red and orange paralenes. It's worth noting that in this case, we add the ligands after synthesis in a post-synthetic ligand exchange. So we start with cesium lead bromide nanocrystals, we then add our ligand and replace the oleic acid and lailamine on there with these molecules. So what that does is effectively destroy the nanocrystals slightly. It would have been nice if we could have maintained their morphology, but you see that we go from a nice cubic structure to kind of this slightly amorphous round structures, but these are still crystalline and they have the same optical properties because these nanocrystals aren't really quantum confined. They're more compositional controls the energy levels. So it's not ideal in that we wreck the nanocrystals a bit, but they still sort of behave like we'd want to see. Looking at the emission, you can see in green we have the cesium lead bromide. So maybe they've, and black is the mixed solution, and then orange and red are different ligands. So the first thing we see is in the mixed solution, there's the question? No. In the mixed solution, we see that it's blue shifted slightly, and that's probably because these nanocrystal particles are a bit smaller on average. So you've got a bit of quantum confinement there. And then what we then see is we do get emission from our paralenes. It's hard to tell if that's energy transfer yet because it's impossible to just excite the perovskite nanocrystals because you look here between sort of 400 and 500, everyone's absorbing. But then we go into excitation and we look at, firstly, we look at the emission from the perovskite nanocrystals at 510 nanometers, and we see there's this distinctive excitonic peak around 470. Looking then just at the parylene orange case, so just the mixture of these two, we see that if we look at each of the different emission bands of the parylene orange, they all, to different extents, do have this small peak shown here, which is contribution of the perovskite nanocrystals into perylene orange emission. So it means some of the light that's absorbed by the perovskites is ending up and contributing to the emission of the perylene, but not all of it, because we still get the characteristic shape that is the absorption spectrum of our perylene orange. The perylene red one works slightly better, and that's mainly just because you can perceive it better because it now hits into this well. But we see again in both the emission peaks at like 606 and 650, we get this characteristic excitonic peak from the perovskite. So we are getting energy transfer from our shell into our ligand. If we just do an emission map, you can see all excitations and all emissions. You see the perovskite nanocrystals, as expected, emit quite narrowly, but have this sort of semiconductor nanocrystal shape. And then compared to the perylene orange and perylene reds, you do get this sort of slight extension of their absorption or the excitation just based on the energy transfer from the nanocrystals. But it's not quantitative. It's still definitely absorbing its own light. This slide looks a lot busier than I thought it'd be. But the main thing is we did TCSPC and we measured the luminescent lifetimes, and we see what we expect in that the bromide's lifetime is shortened from 3.6 nanometers down to 1.7 and 1 nanosecond, and that's indicative of being quenched. So if you have a FRET process in which energy goes from nanocrystal to your dye or so, the nanocrystal should be quenched. We do see a characteristic increase in the perylene red lifetime, which again implies that it's getting pumped at a later time by this nanocrystal, not the initial excitation. And then when we look at the perylene orange, it gets a bit more confusing and complicated because it's actually shorter when it's attached compared to when it's not. And we attribute this just to the fact that the perylene orange isn't as, if you look here, it's not as sterically hindered as the perylene red. So the perylene red has these big biphenyl positions, and that stops the perylene reds from aggregating and forming dimers. And the perylene orange is quite renowned for forming dimers and excimers. So we found that this is a previous paper we published, that if you attach these nanocrystals to inert silicon nanospheres, so the same sort of thing we did here, but instead of a perovskite quantum dot, we just use a bit of silicon or silicon dioxide, and it has this weird excimer formation. And so we think that's what's going on, and it complicates things a little bit. So there is a bit of energy transfer, but the perylene orange, again, are being a bit messy. So summarizing this first half, we did attach it. It worked. It wrecked the nanocrystals a bit. They didn't come out as nicely as they should, but we did get improved absorption in terms of a light harvesting complex. There's more absorption because now this molecule absorbs a bit of the light from the nanocrystal. There's energy transfer, it's not complete, and the emission has been shifted slightly. The idea for this is that you could also improve the PLQE. If you had a nanocrystal with a bad PLQE and quantitatively transferred all its energy to a dye with a good PLQE, the whole system could have a sort of a close to a unity PLQE if all energetic transfer steps were also unity. But when you still have emission from different chromophores and you can't selectively excite, it gets a bit hard to determine the actual increase in PLQE. So we just kind of left it there and moved on to our second case. So
Nate,
yep.
It's Paul Byrne.
Hey, Paul.
Can I just ask a question?
Yep.
Well, it's two questions. I know we normally wait to the end, but I might forget by then. So my first question is, when you, if you like, etch your perovskite crystals, I mean, one of the things about perovskites is that if you put a large cation, if you like, or into them, then you end up with a kind of you can end up with a 2D structure. So do you know whether the surface of your perovskites start as 3D structures and then go to 2D?
So I think in this case, they're still 3D because the carboxylic acid doesn't sort of bind into the lattice as well as like a thing. So we were effectively in maybe at AUCAOS seminar, if the travel board is down, one of my students has done this, but with an amide linker on a anthracene. And the idea was to make 2D perovskites. And we've got some success in getting that amide to go into the surface and having a bit of confinement in the 2D wells. We had more success starting with a lead bromide and putting the anthracene in, and then creating, and you start with a crystal. If anyone see my hand, it looks like this, and then it sort of does this, and it's like one of the best TM images I've seen, because you see that the addition actually forms a 2D. But for this case, I don't think it has.
Okay, that's cool.
Yep.
And the second question is, when you saw a decrease in lifetime, when you put the organics on the outside, is that always due to energy transfer, or can that also be due to charge transfer? So, for example, in a photovoltaic, you can have channel 1, channel 2. So do you have any feel as to whether it is purely an energy transfer, or whether it can also be due to charge transfer?
So there was a couple of papers where people think it's majority charge transfer, but just by the lifetimes and the transfer process, it seems more to be on the FRET regime. It's probably one of those things where I think it might happen both ways. But I think because we're still getting sort of emission from the nanocrystals and later from the dyes and stuff, I think it is probably more going to be a FRET process, because they're still sort of floating around in solution, and we're not using many, like our fluence is quite low and stuff. So I'd say, we're doing the TA on it now, and the TA will tell us if it's charges, but we just haven't got that data yet.
Okay, thanks very much.
Okay, so case 2 is the opposite to what we looked at. So we're going from ligands on the outside, putting energy in. And this is a photo I stole from one of my papers that uses tetracene and does singlet fission. So ignore the actual shell. But the idea is this absorbs the light and transfers in. And in this case, we did the synthesis with this ligand in the solution. So all the way back up here, where you make nanocrystals, we added the ligand in here during the synthesis. So slightly different. We're trying to avoid destroying the nanocrystal surface. And again, we chose now anthracene because we want to be high enough energy to go into either cesium lead bromide or cesium lead iodide. First thing we see is we maintain a lot of the nanocrystal structure, which is nice. There is sort of slightly here, slight changes in morphologies, like there's a bit more sort of elongated rods than I would expect from a normal synthesis. Normally you can get sort of quite good, very monodispersed cubic ones. And then our iodides slightly get a bit bigger and rounder. So ligands are known to have structural dictating effects during synthesis, so it's not too surprising that that happens, but these still look a lot more like nanocrystals than the last case. And then again, it's just a very simple story, as if we look at the absorbance, we see that compared to the standard nanocrystals in both the bromide, or the bromides over here and the iodides, we do get this anthracene peak, which means there's anthracene at least attached because we've washed the nanocrystals, and you can see that these match with the absorbance of our anthracene molecule. In this case, the emission is slightly shifted, but we see no anthracene. So in this case, it looks like we are, at least for this purpose, getting quantitative energy transfer, or just destroying the anthracene's emission through dimers on the surface somehow. But there's no anthracene emission, and we get our 2 peaks here. And it shifts slightly. This shifts probably just due to the different size. And same with these ones. It's probably a nanocrystal structural effect. The excitation shows us there are slight contributions from the anthracene. It's hard because nanocrystals absorb so much because they're semiconductors. So we get these 2 sort of bumps here. And sometimes they correlate slightly negatively because there is some anthracene absorbing light that I think is going dark. So that cuts out, but there's also is a bit of a positive increase in some of them here as well, which it's hard to know if the shift is because, if we go back up here, you see there's a slight shift in peak heights due to the absorbance of the anthracene when it's attached and when it's not attached. So that means it's implying that maybe there is some form of dimerization or aggregation occurring on the surface. So then when you look at the exotonic peaks, we see like a negative contribution from pure anthracene. So maybe there's anthracene that are dark states, but we see a positive based on the absorption. So maybe some of the aggregates are. So right here, it's kind of like a maybe yes, maybe not case. So then we again go to fluorescent lifetimes. And what we see is when we attach the anthracene, we couldn't do the anthracene emission just because there's none. So it's hard to quantify, but the nanocrystals' lifetimes increase in both cases, indicative of some form of energy transfer. And again, we're doing the TA on this one as well to work out exactly what that energy transfer is. But it seems quite simple and sort of the lifetimes of FRET, but it could be charges, as Paul said. So in summary for this case, we sort of maintained a lot of our nanocrystal structure. We did get our energy transfer into the core, and we can shift the emission of the anthracene all the way out. So now, in terms of a light harvester, the anthracene is absorbing more light, and that's getting transferred in the core. So these are kind of there, but not sort of 100% there yet. We sort of are getting up to these stages, but the energy interactions of both these cases weren't sort of ideal. So in the future, it's then now we sort of prove the idea and the concept. We go through and find ideal nanocrystal to sort of try, and then we change our binding group to control the density or the numbers or the strength of this binding interaction. We change the linker to sort of stretch out the energetic interactions between the particles here, and then we go for different chromophores. And so I think there's a lot of scope and research there. And we're also going to explore how the difference between synthesis or ligand exchange affects things. So in summary, I'd just like to thank everyone I worked with: Mike Price, Ellen Rossi, Jay Carty, and Matt from Victoria, Timothy Schmidt from UNSW, who was helped me synthesize these molecules back in my honors year, and Jeff Gorman from Cambridge, who turned those molecules into a carboxylic acid. If people wondered why there was such a weird group on the end here, it's because we had an alcohol to start with. And then just funding from the Marsden Fund, the Ministry of Innovation, Business Innovation, and National Science Challenge. And thank you everyone for listening.
Excellent. Thank you very much. That was a really, really interesting talk. So we'll allow people to ask some questions. Actually, can you see chat, Nathaniel?
Can I see?
Okay, so we have a question. Jos Kistermarker says, can you use fatter linkers to spread the chromophores physically further from each other to prevent aggregation?
Yeah, so that's one of the things we're looking at. So using sort of like either down here a bidentate or tridentate linker to take up more room. It also has the problem of we're trying to do the opposite, is because nanocrystals need so many surfaces bound. So with lactic acid, there's about, I think, 3 per nanometer squared, whereas this ligand here only has 1 per nanometer squared. So that's really good for the ligand, but you've wrecked your nanocrystal. So it's a trade-up between nanocrystal surface passivation as well as ligand interactions, if that makes sense. Just trying to work out how I see chat.
Sorry, he says thanks.
Cool.
Were there any other questions from anyone? Just remember, you'll have to unmute your microphone.
Well, then I'll ask a question.
Yes.
Thanks for the talk. Your nanocrystals seem to suffer a little bit from this. Is that surprising? I guess this is, I think Paul kind of touched on this a bit. This is due to the perovskite hybrid nature of the materials. So I guess it wouldn't happen if you had a more robust nanocrystal. So are there any strategies that you have in mind for enabling you to do this on these sort of softer perovskite materials?
Yeah, so in my experience with all the lead sulfides, they're easy. Nothing wrecks them. They're really good. I've got a student who's making, effectively, they're like bidentate, but a long linker ligands. So you have one ligand here, and then it's a carbon chain, and then another linker, and then that goes off. And there's some good research that shows that improves the stability of perovskites quite dramatically, because one sits in one of the sites, the other sits in the other side, and also structurally holds it together because it's linked. So we're in the process. I think we're about a week away from having some of those ligands that will stabilize perovskite crystal structure. Perovskites also have the problem that the ligands are quite dynamic. I always think of them as like they're not really on. They sort of move in and out quite regularly. And this is meant to be quite a strong binding affinity. So I think that will not only increase the stability, but also allow us to be a bit more confident in what's happening on the surface.
Okay, thanks.
All right. I think we might have to leave it there in the interest of time. So everybody, please join me, kind of virtual round of applause for Nathaniel, for our speaker.